I'm Calvin Montfort. This is first of my pharmacology videos. It's on pain. Pain is a nervous stimuli mediated by chemical messengers to nerves. It could be due to inflammation, lactic acid accumulation, nerve irritation, or severe vasoconstriction. You see, nerve is transmitted by pain fibers of the nerves. Uh, these pain fibers should have some chemical irritation by inflammatory cytokines in case of inflammation, and lactic acid in case of fatigue and anaerobic respiration. Pain pathway. The pain pathway. The pain pathway starts in areas like skin, like cutaneous parts of the skin, and GIT. GIT, there are less pain fibers, and the skin there are more pain fibers. So this dorsal pathway, signated by yellow, is a faster pathway in the posterior part of the spinal cord. This is the cut section of the spinal cord, and this is posterior, and this is anterior. This is anterior, and this is posterior. So cut section of the spinal cord, and the faster fibers are at the dorsal part of the spinal cord, and the slower fibers are on the lateral and the anterior part of the spinal cord. So when you cut the nerve fibers, when you cut a nerve, there will be multiple different types of fibers of different type of thickness, like in micrometers. 13 to 20 micrometers is A alpha, the thickest fibers. A beta is 6 to 12 micrometers. A delta is 1 to 5 micrometers, and C fibers is 0.5 to 1.5, 0.2 to 1.5 micrometers. So A alpha, they carry proprioception, they carry position sense, whether we are standing, sitting, or looking up, looking down, raising the leg, putting it down. A beta carries touch sensation. If somebody is touching, it is myelinated, and A delta, it's pain, and in pain it's a faster sensation of pain, and C fibers are slow fibers of pain. They carry slow pain, and A delta carry faster pain. So I've explained this. So when a nerve fiber of a squid, in the squid experiment, a nerve fiber of a squid was taken and its voltages were studied, and multiple fibers of different thickness were found out, and the function of each thickness of fiber and the voltage of the fibers are classified based on Erlanger-Gasse classification. Erlanger-Gasse classification, what it does is it it divides nerves based on the thickness of the nerve fibers, and based on the thickness of the nerve fiber, the function of the nerve fibers determined. A alpha to A delta, the nerve fiber thickness keeps on decreasing, and B and C, the nerve fibers are small fibers. A delta carry fast pain, and C fibers carry slow pain. So here are spinal nerves. Spinal nerves. So higher voltage is A alpha, and C has the lower voltage, but it has a higher. It has the higher. It has a longer period, but has lesser amplitude. So this is a complicated diagram of how a nerve fiber senses pain. First, there is a damaged tissue, like a damaged skin, damaged something, damaged skin or damaged eye or damaged muscle. That muscle will release adenosine, that will release proteinases, that will release nitrous oxide. If it's an artery or a vein, it will release glutamate and various such substances. These substances stimulate various receptors of the nerves, and matrix metalloproteinases are released to the macrophages, and macrophages release tumor necrosis factor, interleukin 6, etc. And the mast cells are the ones which are damaged by substance P. Substance P is the pain substance, one of the major pain substance in peripheral vascular diseases. and this entire thing is the ending of a synapse of a neuron or the ending of a neuron, and these are various immune cells which send out chemical signals of pain that are stimulated by that stimulate the synapses of the neuron. So the beauty of this picture is that if you find a chemical, if you discover or if you synthesize a chemical that if you synthesize a chemical that could bind to any of these receptors and inhibit them, then pain can be stopped. So these can be used for the development of antagonists. It's going to be a complicated substance. Some of the substances invented for this are opioids, non-steroidal non-steroidal anti-inflammatory, inflammatory painkillers, and steroidal painkillers and opioid analgesics. So opioid analgesics work on mu receptors. I hope I find mu receptors here. Yeah, here's the mu receptor. And non-steroidal anti-inflammatory drugs, they work on damaged tissue cell membrane, and corticosteroids also work on damaged tissue cell membrane. So these are the chemical mediators of pain. Whenever these chemicals such as prostaglandin substance P, interleukins and leukotrienes go and stimulate these neuroreceptors such as bradykinin, histamine receptor, serotonin, and all of the receptors, they can cause pain. So what is an anesthetic? An anesthetic is any drug that causes loss of sensation. An means negative, aesthetic means sensation. Hyperesthetic means hypersensitive. Anesthetic means loss of sensation. So an ideal anesthetic should be rapid and pleasant on induction, intravenous or oral or whatever. Rapid changes in the depth of anesthesia. It's too deep into anesthesia. I don't know what it means. Adequate muscle relaxation. Yeah, it shouldn't be when you cut open the body, the muscle shouldn't go into contraction, which will make the surgery more difficult. Wide margin of safety. It should be considering patient safety and absence of toxic and adverse effect. There's no single drug that can do this, so we can use a combination of drugs for anesthesia. That's why we need an anesthetist. So when you look at paracetamol is a non-steroidal anti-inflammatory drug. Here's the chemical structure of paracetamol, and here's the metabolism of paracetamol. Paracetamol is also known as acetaminophen. Acetaminophen is urinarily excreted via urine by glucuronidation of sulfation, and it goes into the pathway of hepatocyte necrosis through NAPOI. It's a little deep into biochemistry. Now let's look at the damaged cell membrane that I was talking about in the previous diagram. The damaged cell membrane the intact cell membrane structure look like this. This is a hydrophilic end, and these lines are hydrophobic ends. When the cell membrane is damaged, cell membrane are lipid attached to phosphate molecules. When lipids are damaged, lipids change their structure into arachidonic acid. Arachidonic acid change their structure into prostaglandin G2, and prostaglandin G2 converts into prostaglandin H2. And this conversion of cell membrane phospholipids into various prostaglandins and arachidonic acid is by phospholipase A. So corticosteroids like dexamethasone, prednisolone, etc., which are synthetic analogs of adrenal cortical mineralocorticoids or glucocorticoids, they inhibit phospholipase A2, and conversion of cell membrane into arachidonic acid does not happen, thus inhibiting the entire inflammatory soup of reactions. If you want to use non-steroidal drugs like ibuprofen and paracetamol, they work at COX-2 inhibitor. COX-2 is another enzyme which converts arachidonic acid into prostaglandin G2. So if you inhibit COX-2, The further inflammatory response does not happen. I would like to explain antagonist in detail and agonist in detail. What is an agonist? An agonist is a drug that binds to a receptor and stimulates the receptor containing cell or enzyme. An antagonist binds to the receptor containing cell or enzyme and does not do anything. It just binds and blocks it. That is the antagonist. So these drugs are antagonistic drugs. And here's a picture of the cell membrane. I'll give you a better picture. Yeah, here it is. Here's the cell membrane, phospholipid bilayer. If you cut it and look at the chemical structure, here's the phosphate group, and here's the hydrophobic ends. Yeah, here's the hydrophilic end. Hydrophilic end. It can attach to water. It can get stuck to water. This part is away from water. So it's a stearic acid fat molecule. So this can get damaged. This can get damaged to release arachidonic acid, which is produced by, which is enzymatically enhanced by phospholipase. And again, here's the picture. Phospholipase A2, it converts cell membrane into arachidonic acid. It converts into prostaglandin G2 and prostaglandin H2. Various prostaglandins have various functions. They can stimulate inflammation, they can cause pain and stuff. And here's the lipooxygenase pathway. Cyclooxygenase pathway is inhibited by non-steroidal anti-inflammatory drugs. Lipooxygenase pathway, arachidonic acid is acted upon by two enzymes, cyclooxygenase and lipooxygenase. Lipooxygenase produces leukotrienes. These leukotrienes cause havoc in conditions like asthma. So leukotriene antagonist can be used. Lipooxygenase antagonist can be used in asthma treatment. Here this is generalized inflammation and pain. So substance P. Substance P is one of the chemical mediators. Here's the molecular structure. It causes pain in arterial diseases, and there is vasoconstriction and damage to the blood vessels. Substance P causes pain. It irritates the neurons. So here's a picture of antagonist. Arachidonic acid goes and binds to cyclooxygenase, but instead of that, paracetamol goes and binds to arachidonic acid analog, which is paracetamol. It goes and binds to cyclooxygenase and just blocks it. It doesn't do anything, doesn't stimulate it. And this arachidonic acid, which is secreted, it goes into the urine or feces or whatever. It gets metabolized. So again, here are the chemical mediators of pain on nociceptors. Excess potassium can cause pain. ITP, ADP can stimulate the nerves. Bradykinins, leukotrienes, like leukotrienes of lipoxygenase pathway. Serotonin, histamines, prostaglandins. So excitatory stimulators of pain are substance P, calcitonin, glutamate, aspartate and ATP. These bind to the neurons and stimulate them, causing pain. Inhibitory stimulus are somatostatin, acetylcholine. Okay, these drugs work and inhibit the central nervous system pain response. So thanks. I would like to elaborate more in detail, but I've taken pictures from the internet. I'll use this as a reference, and I'll add a video with my hand-drawn diagrams on it. So thanks.